We now turn to a scientist whose genetic work in the model organism, the worm known as C. elegans, has illuminated one of life's most fundamental signaling pathways. Dr. Iva Greenwald has provided key insights into the Notch signaling pathway, one of biology's most conserved systems for cell communication and cell fate decisions. Her discoveries have shaped how we understand development, tissue maintenance, and even cancer. Please welcome Dr. Iva Greenwald, who will speak on LIN-12/Notch and the awesome power of C. elegans genetics.
I'd like to start by thanking, I'm sorry, I'll be like this for a minute until I get going, the Gairdner Foundation for this overwhelming honor. And honestly, having heard the talks this morning, I'm even more overwhelmed and honored and truly humbled to be part of this endeavor. So I'm going to start by giving you an overview of the novel mechanism of Notch signaling. And I'm hoping my pointer, good, that's why part of the reason I'm down here. Notch is a transmembrane protein that's essentially a membrane-tethered transcription factor, where the transcription factor part is in the intracellular domain. Binding by ligand creates a pulling force that exposes a cleavage site for an atom protease that clips here and leads to ectodomain shedding. And that creates a small transmembrane stub that becomes a substrate for a multi-enzyme complex called gamma secretase that clips here and then releases the intracellular domain to go to the nucleus and activate target genes. And between Gary and me, you'll hear much more about these steps. So I have organized my talk using a slide that I showed to my undergraduates at the end of a unit on genetic approaches to biological problems, because my work has really been following this canonical flowchart, starting from the top and going to the bottom. It's been a privilege to be so close to each of the steps. And so let's talk about what the process of interest is. It's cell fate specification during animal development. And we all know we come from a single-celled zygote. And a common theme in animal development is that as the cells are proliferating, groups of cells are endowed with a certain developmental potential. We call those equivalent cells. And that means they have some choices to make about what they're going to do. So many of these choices are made when they get signals from outside the group of cells. That process is called induction, and Notch mediates those types of interactions too. Notch uniquely mediates a type of cell interaction called lateral specification. And this is when you have a group of equivalent cells and they signal amongst each other so the correct numbers and types are made. And you'll hear this generally referred to as lateral inhibition. And I'm going to show you an experiment soon that will explain why I prefer the term lateral specification. Okay, so the screen. Bob Horvitz did brilliant genetic screens to open up many different biological problems in C. elegans. And one of the screens he did was to identify viable and fertile mutations altering vulval cell lineage. And Bob actually did this himself. He pioneered the screen. And then my wonderful classmate, Chip Ferguson, expanded this on an enormous scale. And the screen was so effective because here's a wild-type worm, and it shows the vulva. A C. elegans hermaphrodite is essentially a female that can make sperm, and she can make self-progeny through internal self-fertilization. So if an animal doesn't have a vulva, she doesn't need to mate to make progeny, and it turns out she can make progeny by self-fertilization, but the eggs can't be laid. And instead, they hatch internally and eat their way out. They're only a millimeter long, so it's not as gross as it sounds. But you can see here, it's a very distinctive phenotype that you can see on a Petri dish, and we call it a bag of worms. There's another phenotype that you can see that affects vulval development. I don't know how, I can't look up. These are visible pseudovulvae, and those turn out to reflect aberrant signaling in cells with the equivalent potential to be vulval cells that normally aren't. So Chip identified a number of mutations that were dominant, mapped to a small region of a chromosome, and were either vulvaless. Well, they were all vulvaless, and some of them were also multivulva. And that's where I came in. I did much of my thesis work on something else, but I had actually joined Bob's lab to do development. And so I had my pick of genes at that point, and I chose LIN-12 because I was intrigued by these different dominant mutations. And my previous work had prepared me for how I could use dominant mutations to analyze processes. So I started by characterizing the mutations in the genes they define, the nature of the morphs, and time of action, which I won't talk about. And I didn't do this alone. That's been a theme today. We hear about how we have colleagues. And I had the great good fortune to have an extraordinary scientist and a wonderful person, Paul Sternberg, work on this with me. And as we published this paper, and I'm calling it Eureka Moment 1, I'll label some of these when we felt we learned something important. And as came up earlier, I mean, sometimes you see something amazing like the cilia, but sometimes you build a lot of things by putting them, you put a lot of things together and build that Eureka moment. So students, don't worry if you don't feel that way after every experiment, but hopefully you'll feel that way sometime. So we concluded that LIN-12 is a binary switch in many different cell fate decisions. And what I mean by that is if cells have the potential, say, to be A or B, and one of the fates is associated with high notch activity and one is associated with low, it's the low versus high, on versus off nature of that notch switch that specifies their fates. And we know that because I was able to identify null alleles of LIN-12, and both cells became fate A. And the gain of function, dominant alleles turned out to be genetic hypermorphs or elevated activity alleles, and both cells became fate B. And we found this was true in many different cell contexts, but each cell context offered a different binary choice, not A versus B, C versus D, stem cell versus differentiated cell, or I'll show you a real context in the worm later. And an important thing that was, quite wonderful timing at the time is that we knew at the time even that at least some of the decisions involved cell-cell interactions to resolve. So as we move down our flow chart of how to do a canonical genetic approach, the next task was to clone and sequence the gene. And I did that as a postdoc. And I reported the cloning and partial sequence of LIN-12 back-to-back with a paper from Spiros reporting the full-length notch sequence from Drosophila. And you can see from the title that EGF repeats was a big deal. And I should really give you some context again for the students. This was in the 1980s. We didn't know a lot. And so finding even a little bit of mammalian homology was very exciting, even if we didn't know quite what it meant. And indeed, the only developmental switch genes that behaved in this binary way, UBX and Antennapedia, that had been sequenced at that point, were Hox-containing nuclear proteins and likely transcription factors. So the presence of EGF repeats indicated at least some role involving the outside of the cell and potentially in cell-cell interactions, which was exciting to me because of the genetics in the worm. So we actually knew more sequence than we reported at the time, thanks to a wonderful student, Kathy Weston, who got intrigued and did a lot of sequencing. And then John Yoakum not only finished and cleaned up the sequence, but cloned a second C. elegans notch gene. And we, the community was thinking of, well, how does this transmembrane protein mediate cell fate choice? And, uppermost in our mind was how does it mediate cell-cell interactions? And so because of the binary nature of the switch and the sequence, we were particularly focusing on instructive cell-cell interactions, whether it be information that it's either a signal or a receptor. And there were other hypotheses as to the function of this membrane protein at the time that I won't go into anymore. So the next question then, if you want to know if something's a signal or a receptor, you want to know, does it function in the signaling cell or a receiving cell? And as a geneticist, our approach of choice is mosaic analysis. My brilliant student, Geraldine Seydoux, did this mosaic analysis, which was only just becoming able to be done at the time. And, and we reached two conclusions from our study for reasons I'm going to take you through in more detail than anything else that we've done. So we concluded that the simplest hypothesis to explain everything is that LIN-12 is a receptor. But the point I'm going to amplify on, no pun intended, is that these cell fate specification events that involve lateral signaling involve feedback amplification of a small initial difference in notch activity. So the paradigm that we use a lot and has been so revealing for this particular point is just two gonadal cells that interact with each other and resolve which one is going to be an anchor cell and which one is going to be a ventral uterine precursor cell. Now on the right here, you can see that when an L1 larva hatches, its gonad has only two cells, Z1 and Z4, that will make the entire rest of the 155-cell gonad. And that Z1 PPP and Z4AA, which I'll sometimes call the alpha cells collectively, these names are the lineage history of those cells, the posterior daughter of the posterior daughter of the posterior daughter of Z1, et cetera. So these cells come from different lineages, but they come together and then undergo this process to decide which one will be the AC, and there's always only one AC, it's perfect every time, or which one will be the VU. So LIN-12 is and I'm using LIN-12 and notch interchangeably. LIN-12 is a binary switch in this decision, so 50-50 in wild type, but in a null, both become ACs, so you know you need LIN-12 activity in one of these alpha cells to be a VU. And the hypermorph, where you have elevated activity, high LIN-12 activity causes the alpha cells both to be VUs. So Geraldine's experiment was simply to juxtapose a cell that lacked LIN-12 activity to a cell that was wild type. And what she found when you look at the LIN-12 null cell is it always became an anchor cell. And that says that LIN-12 is acting cell autonomously in the receiving cell to specify VU fate. And that was important at the time because that pointed to a role as a receptor. But I didn't label this eureka moment because this is the eureka moment. So we were very surprised and both puzzled and energized to figure out what it meant that the wild type cell always became a VU. In an intact animal or an intact wild type cell, it has a 50-50 chance, but in this mosaic, when this cell had to be an AC because it couldn't receive a signal, this cell always became a VU. And the importance of this is it says sorry, that the cells are communicating with each other before either commits to a fate, to coordinate their cell fate decisions. Otherwise, you might have expected this cell, the wild type cell might have 50% of the time decided to be an AC, tried to inhibit the other and couldn't because it didn't have the receiving possibility. So that's why I like lateral specification more than lateral inhibition, because I like to emphasize the conversational nature of this process. So we proposed, and kind of remarkably, what we proposed has been substantiated, that initially both cells are expressing LIN-12, and the ligand, which wasn't known at the time but is now known to be a gene called LAG2, and that a small stochastic initial difference that would give one cell a little bit of a head start with LIN-12 would become amplified by feedback mechanisms so that the net signaler would have less receiving and the net receiver would have more receiving, and eventually they would commit to the AC or VU fates. So a couple of years later, Pascal Heitzler and Pat Simpson published an absolutely gorgeous genetic mosaic analysis in flies, a particular fly paradigm that was very good for this kind of cell-by-cell analysis, and also concluded that feedback amplification of initial differences in notch activity was important. And this was really great because when you have similar kinds of things in two very different experimental systems, even if you have multiple examples in one, but if you have it in two very different things like worms and flies, you could be more confident you're onto something fundamental. So great students, Hillary Wilkinson and Kevin Fitzgerald, who you'll be seeing a bit more later, were pushing the edge of what kinds of reporters we could do in the worm at the time. They fused beta-galactosidase coding region to regulatory sequences from LIN-12 or LAG2, the ligand, and found, and just let's look at this point here, I should look at it too, that you could see that at this point, which we know by timing and anatomy is when the decision is occurring, only one of the two cells expresses LIN-12 and one of the two cells expresses LAG2. And Hillary was also able to show that there was positive autoregulation of LIN-12 transcription mediated by an upstream element. So it was positive autoregulation. So I just want to, and Pat and Pascal Heitzler subsequently also found evidence for a feedback mechanism that at least involved inhibition of ligand expression through a transcriptional cascade. So I just want to stop here for a moment and say that up till now, I think we can, even from these older studies, but certainly from lots of work that's been done since then, can make some general comments about the decision-making logic. The decision itself is pretty simple, notch on, notch off. But what makes it more complicated are the context-dependent mechanisms for establishing the binary choice, for creating an initial difference in notch activity between the cells, and the kinds of feedback mechanisms that are invoked. They often involve both negative and positive feedback, negative to oppose the alternative fate, and positive to promote notch activity in the associated fate. So we've worked out some details of this, and I want to really highlight just an outstanding graduate student, Xantha Karp, a proud graduate of the University of Toronto, who came to my lab to do her PhD work. And she really cracked open the problem of the details by implicating a transcription factor called HLH2, which is the ortholog of Drosophila daughterless and mammalian E proteins, including E2A, which can actually substitute for HLH2. And she established that HLH2 is involved in the binary choice, and she and another outstanding student, Maria Sully, worked out involvement in the feedback mechanism I'll show you in a moment. And this group of wonderful people have more recently using some of the phenomenal tools that I only wish had been available 30 years ago, but they weren't, but they are now, and they could begin to work out how HLH2 contributes to the initial bias that gets amplified. So what we know about feedback from the work of these students is that LIN-12 is positively autoregulating its own transcription, also positively autoregulating transcription of a gene called LAG1, which is the transcription factor with which LIN-12 intercellular domain will interact to promote target gene expression. You obviously want them both up because you want to get those target genes going. And what this, and Catherine Lowe had implicated LAG1 here. And these three implicated the degradation of HLH2. So it's not a transcriptional cascade, but activating LIN-12 directly, well, through the activation of ubiquitin ligase causes degradation of HLH2, which you need to drive the ligand LAG2 at this point. So if you don't activate, if you degrade HLH2, you don't activate LAG2, you're less likely to send a signal to this cell, so you're less likely to activate LIN-12 and less likely to degrade HLH2. So that's the basis for this feedback mechanism. So feedback amplification for notch also is involved in another cell type where it also represents crosstalk with EGF signaling. So in this paradigm, and an outstanding student, Andrew Yu, worked all this out, These cells have the potential to be either a type called primary or secondary, and initially both express EGF receptor and Notch, but after feedback and crosstalk, only one expresses EGF receptor and one expresses Notch. And here the feedback is a different mechanism. Activating LIN-12 causes transcription of a set of genes that act as negative regulators of EGF receptor signaling, which is needed to promote the primary fate. And the positive feedback includes the autoregulation that I mentioned, but also involves the transcriptional activation of a microRNA that is needed to downregulate the GEF, VAV1, which is a negative regulator of LIN-12. And that one I'll show you again one more time briefly later. Okay, so now we go to structure function studies and begin to get into mechanism. So by structure function studies, you know, at our level of geneticists, I mean learning something about what you need for the proteins to function, and that is generally done by sequencing mutations, and at the time many groups, including Spirosis, were generating truncated forms. And Gary and I began our first teaming up at this point with a very targeted kind of truncation that I'll describe that led us to propose the cleavage model. So, but to talk about like the last kind of like real solid piece of work I did with my own hands, I wanted to sequence the hypermorphic alleles of LIN-12 because there are a lot of different ways you can make proteins work less well by mutation, but there aren't too many ways to give them increased activity. And when I sequenced them, and that was not like sending it out to be sequenced at the time. I mean, you can't believe how much work it used to be. But I won't go on that digression. Just don't feel too sorry for me, because at least I got a good result. So what we found was that all these mutations map to the region called LIN-12 notch repeats, or between the LIN-12 notch repeats and the membrane. And we now know, and Gary's going to tell you a lot more about this later, that this is the region that acts as the force sensor for unfolding the ectodomain for the clips. And we concluded, though, and I have a little quote here from the paper, that that maybe the LIN-12D mutations were inactivating a negative regulatory domain. So that if you extend that, then the role of the extracellular domain is to inhibit the intracellular domain, which isn't normally what you think of receptors, right? You think of it as being there to mediate signals that activate something. And that prepared us, or it galvanized us when this very important paper came out from Jeffrey Sklar's lab. It was important for two reasons. It was the first link to cancer, and it was an important clue to mechanism. So in this paper, they showed that a subset of patients with T-cell acute lymphoblastic leukemia, which I'll call TALL, have a fusion that puts part of the Notch1 coding region of humans under the control of the T-cell receptor beta transcriptional control. And this is a figure from their paper, and this arrow shows where the breakpoint is. So this would remove the whole EGF region, but it would leave what I just showed you seemed to be a negative regulatory region, but it would also remove the signal sequence, so you wouldn't expect this to be in the membrane. And so we reasoned if the role is to of the ectodomain is to inhibit the intracellular domain, and this truncation caused cancer by an unknown mechanism at that time, that maybe if we just express the intracellular domain, which would be the only part that would normally be in the cell, that maybe we'd see some constitutive activity. So Gary and I teamed up, and we teamed up for two reasons. One was that it wasn't clear we could even do this experiment in the worm, given the state of transgenic technology at the time. And the other was, again, this idea that if you show things in parallel systems, that it has more power, especially you're dealing with invertebrates and is extrapolating to vertebrates. And also, I'll be honest, the worm wasn't as accepted back then, as hard as it is to believe now. You know, people were thinking there were weird things about worms, so it also gave some power to boost the worm by, you know, associating with the fly. So I didn't label any of that eureka moment either, because this was really the eureka moment, although really it built to that. I left out something important, which is when we express the intracellular domain in a null background, so it didn't have endogenous Notch or endogenous LIN-12, we got phenotypes associated with activation of Notch. Gary will talk a little bit more later about the flies. I'll just show you that I told you the null in C. elegans has 2 anchor cells, and when we put in the intracellular domain, you get the opposite phenotype. We'll see. Anyway, the eureka moment was that it localizes to the nucleus, and we hypothesized, so Kevin Fitzgerald, of course, was very involved in this work, and we hypothesized that the mechanism of signal transduction involves cleavage and transport of the intracellular domain to the nucleus, and may even be involved in transcriptional regulation. So Gary's going to tell you next about, if I may say, absolutely gorgeous piece of work he did that established many of these tenets of the hypothesis. And in parallel, Rafi Kopan was doing really great ex vivo analysis in cell culture, very complementary experiments that provided strong evidence for the cleavage and release hypothesis. So then the question becomes, what cleaves Notch in the transmembrane domain? Here's where we come back to the awesome power of C. elegans genetics. We did suppressor screens and found, we was Diane Levitan, a great postdoc who was doing these screens actually looking for something else, but came across suppressor or enhancer of LIN-12, number 12, in a screen for suppressors of the hypermorphic alleles. And she did the kind of genetics we do to show it was generally interacting with Notch proteins in the worm, LIN-12 and GLP-1, and that it acts in the receiving cell. And literally, like as we were about to send off the paper, we found from work done here in Toronto from Peter St. George Hislop that familial early onset Alzheimer's disease could be associated with mutations in a gene at the time called S182 and now called Presenilin 1. So that was exciting, but what did it mean? Well, what it meant for the Alzheimer's field was obvious. You know, it was an important thing for them to do, but it didn't, cell 12 null mutants didn't have canonical phenotypes. So we felt like we were on the verge of a eureka moment, but we weren't sure what that moment was going to be until a great student, Xiaojun Li, found that the reason that cell 12 null didn't have a lot of phenotypes is that there's a second functionally redundant presenilin in the worm, HOP1. And here's an example of a result. If you take away HOP1 in a cell 12 null background, you get phenotypes associated with the loss of Notch, which means wild-type worms have a rectum, and if you don't have Notch activity, you don't. That's one example. Also affected ACVU and stuff like that. Okay, so then Gary and I teamed up again. He had all these amazing tools from the study that he did to establish the mechanism. And again, Bart de Strooper, who was a fantastic scientist in the Alzheimer's field, teamed up with Rafi Kopan. And again, not knowing, we ended up with back-to-back papers that established that presenilin is required for transmembrane cleavage of Notch. Gary will tell you more about that too. So Xiaojun did something else that I think was really important at the time. Presenilin was, deduced to be a multipass membrane protein by sequence analysis, but it was typically drawn as a 7 because everybody was thinking things like G protein coupled receptors. But what Xiaojun did was he used an in vivo topology approach and identified a spartle containing transmembrane domain. So here we were with a multipass membrane protein with a couple of aspartates. And the biochemists who understood this realized that this could be an aspartyl protease active site, and they proved it by getting transition state analogs that bound there. So what do Alzheimer's and Notch signaling have in common? Well, one thing is the parallel proteolytic processing events. In the case of Alzheimer's disease, an associated factor is the deposition of beta amyloid plaques, and that comes from the cleavage of a transmembrane protein called beta APP, ectodomain shedding transmembrane cleavage, and in this case, this is the focus of much work in that field. In the case of Notch, there's ectodomain shedding, transmembrane cleavage, and signal transduction. And Gary will show you how he demonstrated that any transmembrane protein with a short ectodomain can be a presenilin substrate. One last slide about the awesome power of C. elegans genetics, which I haven't done justice to here for time. But the suppressors of the activated LIN-12D hit pretty much all the core components. When you have an activated protein and you knock down things downstream, you can reduce the hyperactivity of that protein, and so we got a lot of other things. But I think presenilin was probably the star of the show from that analysis, even though the other things were important too. I also just want to point out that from looking at suppressors and enhancers, we've also identified negative regulators that have proven subsequently to be tumor suppressors and sometimes prognostic markers in TEOL. One of these was called cell10, suppressor enhancer of LIN-12 number 10. And its ortholog in humans is called FBXW7. It's a ubiquitin ligase that targets the intracellular domain for destruction, thereby increasing activity, which is how it contributes to TEOL. And VAV, which I mentioned as part of this crosstalk, a negative regulatory circuit, has also been implicated as an important tumor suppressor in TEOL. You also get things involved in general cell biological processes because membrane proteins are trafficked, they're modified, quality control, et cetera. And sometimes they're more illuminating to those processes than the processes are to Notch. But a challenge is always to sift through and find, what's integral, what's core, what's potential modulatory, because any of those things can be potential therapeutic targets. So my literally final thoughts, I keep building a crescendo and then coming back, is that why do you care? What's the basic understanding, especially after today, like these extraordinary talks with, fantastic human import. Sometimes it takes a little longer to get there. Notch proteins play conserved roles and mechanisms in cell fate specification in all animals. They're continuously functioning, not just in development, but also through life in regulating stem cell homeostasis. They're, as I mentioned, can be oncogenes like in TEOL. They can also be tumor suppressors themselves, so everything gets flipped around. And of course, these things can be potential therapeutic targets. Gary's going to tell you later about about ways that the mechanisms and the approaches he used to deduce are being repurposed for some fantastic new tools that will be what you young people may end up doing quite a lot of work on too. Okay, do I have any time? Half a minute? Half a minute. Too many people to thank. I'm sorry, but I just need to thank first and foremost my lab. The theme today has been about teams. I've had just incredible fortune to work with people, and I made some joyful discoveries myself, but it's even more joyful when you do it with your trainees. And I want the trainees to know that, because it's just a special thing when it comes with that mentor-mentee bond. They've supported each other through the times when you don't get those eureka moments or things don't work, and celebrated with each other. And even when people didn't work on Notch, as some of these people didn't, about half, we've all been together as a team. I want to thank the worm community. The worm community is a very special place. The sharing of unpublished information and approaches and reagents is just an important part of everybody's story in our field. Of course, Gary, I don't see him. Ah, good. Got a little worried there for a moment. I owe everything to that's good in my life, and stand up for science.